connecting to the...
Thank you. If I say that, record on this computer, it says, please ask the host to give you permission to record. Do you have something come up that says that?
No, it just says it's recording now.
Okay, all right, we'll trust it then. Hope for the best. Thank you.
Oops. Sorry.
That's all right.
Going everywhere.
All right. Welcome, everybody. I'd like to begin by acknowledging the traditional owners of the lands on which we're meeting today. I'm on Wurundjeri country, and I pay my respects to elders past and present, as well as to any traditional owners who might be joining us tonight. I also pay my respects and acknowledge the traditional owners and elders past and present of the lands from wherever you are streaming in from today, be it Australia or elsewhere. I pay my respects to the First Nations of all nations around the world. So tonight is the last of Frogs Victoria for 2021, which is quite sad, obviously, but also quite a joyous occasion because we are making the most of COVID restrictions and having the last of our interstate speakers. And I have a delivery arriving, so my dogs might be about to greet the delivery driver, and my apologies in advance if that happens. Welcome to you all, wherever you come from tonight. I think we have some new people in the audience, which is always exciting. Welcome to you, and welcome to everybody else that is joining us again for another time tonight as well. I'd also like to welcome, have we got Professor Murray Littlejohn with us? Our patron, Professor Murray Littlejohn, as well. Special welcome to him. Like I said, this is the last Frogs Vic for this year. However, we have a newsletter coming out in the new year. So if you have newsletter articles and or notes, events, things that you'd like to put in Pobblebonk, please get in touch and let us know. We'd like to include anything frog related that you've got to go in there. And the next meeting will be our AGM in March, the first Thursday of March 2022. Now, enough of that. We'd like to welcome, like I said, another interstate speaker tonight, which is very exciting. We have with us Dr. Renee Catullo, who is a research associate at the University of Western Australia now, but that is a fairly new position. Renee did a Bachelor's of Science at Oregon State Uni, which might explain the accent to you, but has also done a PhD at Australian National University and has done lots of work with museums, Australian and Western Australian, and I think museums Victoria as well, but I might be about to scoop the story if I tell you too much about that. So maybe I'll let Renee take over and talk to us all about how many species of toadlets, Uperoleia, are there. Thanks, Renee.
No worries, thank you. Yep, so just turning off my own video so I can't see myself. Yeah, so I'm just thought I would kind of give an overview of a particular genus of frogs. So I started my PhD back in 2008, and I was working on this group, Uperoleias, and it's been an ongoing question. Now I work on many things, but I've never actually stopped working on Uperoleias, and I think they're quite interesting. So I thought I'd kind of give an overview. Everyone hates them and I love them, so I thought I'd make an effort to try and convince everyone that they're actually pretty good. And then talk a little bit, just an overview of what it's like to do fieldwork up north trying to catch frogs. I'll note, I didn't bog this vehicle, which is why I am so excited by it. Sorry, just checking the chat to make sure. So yes, oops. So who are the toadlets? So toadlets are the genus Uperoleia, and they're a really diverse family. They're actually the largest genus of frogs in the Myobatrachidae, which are from Gondwanan, which means that they're potentially up to 100 million years old or more in Australia. And their closest relatives are actually frogs from South America. They are hugely, and I like to say they're the biggest genus of frogs in Australia once the Latoria get fixed. So they're endemic just to Australia as well as the lowlands of New Guinea. And right now there are 29 species of Uperoleia. And most of that is in the monsoonal tropics. So this is a phylogenetic tree of the Uperoleia. So this is showing you the relatedness between different groups of species. And all of the green species are monsoonal species with a nice big clade of mesic species with a few arid species dotted in. These are two species that I have worked on a lot, which are Uperoleia inundata and Uperoleia lithomota. Now, one of my very favorite sort of facts about Uperoleias is they have this bright flash coloration, or most of them do. And if you're trying to say, take a nice picture of a Uperoleia and you give it a little poke in its face, maybe to try and get it to sit up or do something, if you do it very much, what happens is they do this ridiculous little posture to show you their flash coloration. So this is a Uperoleia rugosa. What they do is they shove their little butt up in the air and they stick out one or both legs and show you that flash coloration. My favorite thing about when they do this is they're always one-sided. They're like right-legged or left-legged because they have one leg they're kind of supporting themselves with and another that they're shoving out. And it's always the same one they shove out. This is a really funny one because this is a little female. You can see that eggs actually through her skin, and she actually doesn't have any flash coloration, but she is giving the whole, showing you that she's poisonous, a really red hot go. And it's very cute. You see these little arms, they wrap them around their faces. It's a really adorable and completely non-terrifying display. Now, I actually most recently published this as terms to use when talking about Uperoleias. They're divided into three major groups that I tend to talk about: the eastern squelchers, the monsoonal squelchers, and the monsoonal clickers. There's a few caveats here. There's a couple of this long branch was a couple of Kimberley arid species, which are very annoying and defy my classifications. And indeed, we don't know the call of one of them. And for some reason in this monsoonal clickers, we of course have an arid and a mesic species. So when I talk about clickers, I'm talking about something that sounds like this. Versus a squelcher who is making a sound like this one, which is from the monsoon tropics. Or this is Martini, which is the Victorian Uperoleia. And so I use these different calls as a major identifier for the species. And it's not unusual, particularly in the tropics, to find clickers and squelchers in the same chorus. And I apologize, every call of the north is going to have that honk of the Latoria convexusculus in the background. So yes, so we have these major groups. We can say that probably the whole ancestral group made sounds like a squelch, and that squelch is diversified, but then one clade has evolved this click as a call. Something really interesting about clickers is they've lost this special second call that they have. So this is calls of Uperoleia borealis. Now, this is our waveforms, and this one here on the left is the call that the borealis do in the Kimberley when they're making a call. And it's a very long call. You see that there's a lot of pulses and they're all very evenly spaced. And this is a call that the frogs do when they are just really, really going for it, when they're just in a mate calling mode. But if you disturb them, if something seems to be coming up to them, they do this alternative call. And those calls are slowed down. And what they do at the beginning, though, is have that really set of really, really compressed pulses. And this just means that it makes Uperoleia a lot harder to tell apart, because you'll often hear a chorus of one species of squelching Uperoleia. This includes martini. I have recordings of martini doing the same thing, where they're making this call, but there's somebody in the background doing something different. And that's partially because Uperoleia are quite aggressive with each other, and so they want to warn everybody off of their perfect little amphitheater that they've identified. So what I'm gonna play for you is two different borealis calling. And what you're gonna hear is one guy kind of going normal, and then he's going to make a kind of funky call. And then the second frog is going to come in and do some normal calls and then do a funky call. And then the first guy comes back, does a funky call, and then starts calling. And this is just really common to hear out in the environment. So these are normal calls going through, and there was a short one right there. And there's a really good short one. And now it's dropped in its frequency, and you can hear that it's gone back to its normal call. And there's a really good short one from the first guy coming back in. And now he goes back into a very normal set of calls. And so it's really common to hear these two sets of calls, and they don't necessarily do them as when they're coming in or going out. They'll switch back and forth between them fairly regularly, particularly if they're a little bit disturbed, if they're not really into the chorus yet. I mean, I don't have to tell everyone that Uperoleias are morphologically cryptic. So with that annoying call comes the fact that they all look very much alike. Pretty much every analysis of species shows you that they're all shaped pretty much the same. But there are, particularly in the north, some diagnostic characters. So when you're looking at squelchers versus clickers, those two different genetic clades, one of the key ways I tell them apart is the placement of their inguinal glands. If you look at this here, you see a really lumpy round gland. And when their leg is covered in a squelcher, or a leg is in, basically you can't see this inguinal gland. And that's true for pretty much all the squelchers, even on the East Coast. But these clicking Uperoleia have evolved a different inguinal gland. So you can see this very long, thin inguinal gland. Sometimes it's really dissected. So it looks instead like a whole bunch of tiny little glands, but that's just one long inguinal gland. And in things like rugosa, it's just one enormous gland. But it's a really good diagnostic character without calls to get you to, is it this clicker or a squelcher? Ventral pigment is really important, and I take no credit for this. This was all figured out by Simon Clulow. In Eastern species, if you're trying to figure out Fusca versus Levigata versus Tyleri martini, this is what you need to look at, is their ventral surface. So if you look at this Fusca here, it's got sort of this cream belly, which is really strongly flecked. But the big characteristic is the thighs. If you look at these Fusca's thighs, it's really pigmented. You can't see it through it. Whereas with Levigata, it's got cute little pink thighs. Essentially, even if it has some white speckles on it, you can see through the skin to the muscles, and that is a hugely diagnostic character between the two. Tyleri and martini are very diagnostic because they have this blue-black belly. Sometimes it's a dark gray. I think if you're a colorblind guy, dark gray, but blue-black if you're a lady. And it's just the darkest belly on any frog. The other thing is that these guys have yellow flash, and Martini laevigata have orange and orange or red flash. There are other diagnostic features in other species, like Uperoleia micromeles has a really flat face, like a notodon. Certain Uperoleia russellii has very webbed feet, but pretty much every species that has these other types of diagnostic features is from the arid zone or the Kimberley or the top end, so it's not as helpful. So the rest of identifying it is calls and geography. And those distributions are really important. And so I've used huge amounts of genetic sequencing to figure out where these species are. And I've pretty much given my data to every recent field guide. So the field guides are pretty accurate at this stage. And the truth is, overlap between species is rare. So when we look at the East Coast, I'll show you, we have Martini down here in pink. We have Tyleri down here in New South Wales in yellow. Green along the coast is Fusca. White is Murray Darling, and blue is Rugosa. And really, you only find significant overlap between these species at the the Hunter Valley and around the border ranges, where there's these valleys that cut through from the Murray-Darling all the way to the coast. But through most areas, they're not actually particularly interacting. So if we zoom in here to the Hunter Valley, don't ID frogs in the Hunter. That's my #1 Uperoleia tip. So right here in dark blue, we have Uperoleia monii, which is endemic to this sand plain. Blue, here and here, that's Rugosa. Green is Fusca, and white is laevigata. So within this whole region, 100 kilometers, we've got, 4 species of frog. So that's a bit of a challenge. This map over on the right is my samples of Fusca versus laevigata. There's a couple things. There's a little bit of coming in together along the sort of Great Dividing Range, but you can really see here that laevigata is a Western Slopes species and Fusca is a lowlands wet species. It does go up a bit, but really they don't necessarily like exactly the same types of landscape. And I have no data to suggest that Fusca actually goes south of the Hunter Valley. I'm willing to be convinced of that, but no one's giving me any evidence. So what I've sequenced from Sydney has all been Uperoleia laevigata. Down in the south, Tyleri and Martini. I've not found any overlap between the two. The turnover between the two species is somewhere around Eden. It might actually be the Eden harbor and the sort of mountainous areas through here. So I've got samples of Martini from as far north as Nadgee Nature Reserve in New South Wales. And I've got Tyleri from as far south as Pambula. But where exactly that transition zone occurs is not known. And I think this is a lovely, lovely map because you can see these Rugosa, these blue on the coast at the Hunter Valley and the border ranges, but it really is a Murray-Darling species. And you can really see that laevigata here is really avoiding these lowland areas pretty much for the whole coast region. So distributions are really important to ID. We understand them pretty well. And so unless you're at a range edge boundary, your field guides are likely to be pretty accurate if you can get it to the right major group. So since I've started, I thought I'd just sort of summarize where we've gone to taxonomically. So the first species I described was this Pilbara Toadlet, Uperoleia saxatilis, in 2001. This is one of the easiest ones to tell apart from other species. Firstly, is there rock in the picture is the number one. in the Pilbara, there are 3 species. 2 of them come from sandy rivers, and one of them, saxatilis, likes rocky creeks. And so your first bet is if it's on a rock, it's a saxatilis. But these dark spots on the back are easily diagnostic for this species. And it also has these really reddish glands. That's really common in this species as well, which makes sense when you look at the color of the rocks that are in the Pilbara. So this one is actually quite straightforward. It's pretty common in the Pilbara. The main problem with describing the species was Russellii, which lives in Carnarvon. Nobody was going to Carnarvon. We had lots of samples from the Pilbara called Russellii, but we didn't have samples from where Russellii was described from, which is actually the sandy regions south of the Pilbara. The next big thing I looked at was this whole clicking monsoonal group, and split the ratchetting toadlet, so Uperoleia stridera. So this is split of Uperoleia trachoderma. So basically, this line here is the southern extent of escarpments, and this all becomes very black soil. And that's why trachoderma is called the black soil toadlet. When I call these guys the flat Uperoleia, they are very, very different from everybody else. They are super, super, super thin, which is a thing you would not necessarily expect for a burrowing frog that's semi-arid. It's actually, they're all less than 4 millimeters thick at the sort of this point on the head. So they're very, very, very thin, tiny little frogs. Some of the hardest frogs in the world to catch, because you're trying to catch a frog that is on average about 18 millimeters long, less than half a centimeter thick, in sucking wet black soil clay. It's just the hardest work you can possibly do. So in this split, what I found was the stridera, which is on the west, is acoustically and geographically distinct from trachoderma, which is in the east. And these actually, besides where you are, can be told apart because all of the trachodermas have these little bright red or orange tubercles on their back. A really good example, bright red tubercles on this fellow here, but actually they're totally absent in all the stridera we've examined. They're fascinating though, because they have some of the most enormous flash color I've seen on any Uperoleias. I then got rid of Uperoleia capitulata. So capitulata was described as a rugosa, but with really fat glands from Queensland. And as you saw, I've done extensive genetic sampling, and there's just nothing unique. People from Queensland government went out because this one was a real pain for them, and went to where the type locality was, got samples from there, and they all just turned out to be rugosa. The defining feature was having really, really enlarged glands, but that's just, you know, probably the people who found it were annoying the frog. Their glands were really big. So I'll just point it out. So this is a rugosa here that it's a clicking species. It's a monsoonal clicking species that just happens to live in the Murray-Darling Basin all the way down to Victoria. And so what you see here is this really enormous inguinal gland. So unlike all of the other species on the East Coast, if your frog has this huge inguinal gland on its side, not back here, then you know it's a rugosa. It's really a defining feature. The next one was Mahoney's toadlet, which is a narrow range endemic on sand plains near Newcastle in New South Wales. Basically, I describe this as a north of Sydney martini with a pseudophony belly. This belly is a really defining feature. So it has the blue-black of Tyleri martini, but has this really pseudophony-like pattern. And it is a very narrow range endemic species. And most recently, in fact, only about 2 weeks ago, I just described Gurmel's toadlet, Uperoleia gurmeli. So this is, I think it's valid to say, the hardest to find species in all of Australia. It's described from the Wessel Islands. So if you look at this inset, here's the Northern Territory. And then if you go into this set of islands here, it's actually only these numbered islands or lettered islands, A to G. And it only is known from islands B, C, and D. Actually, yes, that's correct. So it's named for Dr. G because I like his music, and this is Yolngu country. This is under the traditional ownership of the Yolngu, whose primary township is Galawinku. This is a fascinating one because this is a species that's only been seen once in 1993. So the story here is that golden bandicoots were going extinct on the mainland from predation, and so they wanted to put them out on all of these islands to save the golden bandicoots. And they went out and did a whole bunch of surveys and said, oh, look, Uperoleias, I'm sure it's the same, and stuck them in the museum and put the golden bandicoots out there. And the bandicoots are doing phenomenally well. But unfortunately, we just introduced something that is known to eat frogs into a place that has what is turning out to be quite a bit of endemic herpetofauna. So we know that there's also skinks and geckos and things from this area. And the reason it's so distinct is because this is a picture of the Wessel Islands. Unlike the surrounding land, like the Elko Islands, the New England Company Islands are sand islands. The Wessel Islands are basically an escarpment that comes out of the ocean. And this area is probably one of our most important areas of endemism that we're only just now getting a hold of. So we don't know anything about the calls or biology. This is the holotype, which is an adult female. I only have one adult female and 2 juveniles. But at this point, I just described the species because no one's going to go look for it unless we get its name out there. So in terms of taxonomy, there's species I don't know anything about, and also probably I don't believe in as distinct species. The first one is Uperoleia marmorata. So that's the type species for the whole genus. So this name is going to last forever. Its type locality is from Australia. So it's thought that it comes from the Prince River region. There's another species we haven't seen in a very long time called Uperoleia orientalis, and that's from the Alexandria Station in the Northern Territory, which is sort of south of Borroloola. And at the time this was described, that was an enormous portion of the Northern Territory. There's also Arenicola from Kakadu. I'm very skeptical about this because everything people have given me said this is definitely an Arenicola, is genetically and acoustically an Inundata. I have looked at all these specimens, and using my very specific gland approach, I can say they're all those squelching species. And so I'm going to tell you my hypothesis. So I think that Arenicola and orientalis are both Inundata. I think Arenicola was a confusion of that second call type that Inundata does. And orientalis comes from an area that nobody thought Inundata lived in until recently. So I think these are both Inundata. I'm going to show you in a little bit. Inundata is Uperoleia crassa. And I think that Uperoleia crassa, which is from the Kimberley, is Uperoleia marmorata. So my hypothesis is all of these are the same species. Right now, I can't prove these 3 things. So the orientalis and marmorata types are overseas, and we need special genomic methods to get DNA out of them because of how they're preserved. And nobody gives me something that I'm convinced represents what people were describing for Arenicola. So it's hard to prove something when nobody gives you anything you think is real. But I'll show you later, Inundata is genetically crassa. So I think this whole complex will potentially collapse down. So our taxonomy is nearly complete despite those synonymies. There's some hint that there's more undescribed rock species in the Kimberley. and nobody's really properly explored Arnhem Land. I'd be a bit surprised if we didn't find a rock Uperoleia in Arnhem Land, but the East Coast is pretty much finished. But there are things we don't know. Nobody's ever recorded the Tanami Toadlet call. Nobody knows what it sounds like, as far as I can tell. I've done a lot of searching. And we also now, I just added Uperoleia, for which we just don't really know anything about the species, except that it exists. We have learned a lot about the ecology of the species recently, of the genus. They seem to be very, very substrate specific. So this is in the Pilbara. What you can see is this outline, and that's actually where the rocks end and it becomes sand. And we can see these squares are all saxatilis. They live on the rocks. These circles are talpa, which is a close relative, and talpa really sits only in these sandy regions, from Fitzroy Land down to the Roebourne Plain. So in this place, you're really seeing very specific substrate requirements in Uperoleias, which is also another good clue for their IDs. In the Northern Territory, I've seen full community level transitions of species. So you drive 3 kilometers across that black soil escarpment boundary, and you go from things like Trachoderma straight into Lithomota and Inundata. So this is not climate, this is substrate specificity. And we see things like the sandsheet heath toadlet is only likes a very particular type of sand, and that's the same for Monii around Newcastle. We also know that they are pretty resilient to changes in their breeding habitat. They seem like they'll just breed in literally anything, anywhere. And that's really good for them. We also know they are the most annoying species to find eggs. So Uperoleia don't lay nests, don't have nests. Basically, the males and females go into amplexus. And then they swim around underwater, and the female attaches 1 egg and the male fertilizes it. And I've gone out in the field with Marian Anstis trying to find eggs and tadpoles, and she literally just brings a stool, sits down in the water, and carefully picks up every piece of grass, every stick, in order to find the 1 egg that might be attached to it. They're really, really hard to look at as juveniles. But we've learned a lot about their landscape use so far, and they're very different. Certainly nothing like Pseudophrony or Crinia in a lot of ways. So all of the species we've looked at in detail are using a huge area of the landscape around the breeding aggregation. So this is from a study by Martin Westgate. I forgot to put the citation on there, but Martin Westgate, what they did was basically there was a pond, and they put pit trap lines out from that pond on a high relief transect. So that's away from, or that's not on a creek line, and then a low relief transect or along a drainage line. And they put pit traps from the pond out, and they put them on each side of the line. And this is to actually see if they're going towards the water or away. And what they found was basically these overlapping bars showed that out to 200 meters from breeding, Uperoleia tyleri is all mathematically the same. So they are using the landscape relatively equally across hundreds of meters. And they saw no drop off in detectability. That meant that they were basically as likely to find a Uperoleia 40 meters out from the water as 200. So tyleri is using a lot of habitat. I'm part of a study looking at Inundata and Daviesay in the north. They are using hundreds of meters. So in Inundata, we are getting metamorphs, which are the size of your pinky fingernail, more than 500 meters out in pit traps from the nearest place they could have hatched. So it's just crazy how much landscape they use. We also know Monii from Newcastle has been pit trapped 400 meters from the nearest breeding site. And if you look at where Uperoleia disappear from, they're disappearing from areas where native vegetation is no longer connecting to the breeding habitat. So these guys can breed in just about anything, but you have to have quite a lot of negative vegetation near that breeding habitat for them to persist. So tyleri, sorry, I forgot about this. So it uses both high and low equally. So there's no real big difference. They don't mind being out in the forest and not along a creek line. and they show migration. So this study had Uperoleia with Crania signifera and Pseudophorone biberni, and only Uperoleia showed migration towards the breeding habitat. And I think these things are true for most Uperoleia species. They need a lot of habitat, they migrate towards the water, and but they don't really care too much how the quality of the water. I think babies may be different in the Northern Territory, but for most species, I think this is true. So as time goes along, we're getting more Uperoleia being listed. So Daviesii, which lives on really good building sand right near Darwin, is considered vulnerable in the NT. Uperoleia martini is critically endangered in Victoria. And I've done some good bit of survey work for them. And you go to historic sites, and the difference between where they persist and disappear appears to be native vegetation. So they don't care if it's a cow pond, as long as that cow pond has half of it running out into forest. Manii is endangered in New South Wales, another narrow range endemic. And I know that New South Wales is looking at Tyleri. So really, we're seeing declines in narrow range endemic species and near the cities and on the coast. And I think this is primarily due to the loss of habitat around breeding sites. But now that we know things about them, they're actually really interesting species. So I'm not asking you to understand this figure, but this is the types of figures I have to make for all Uperoleia. Basically, standard types of genetic sequencing don't work to ID species. We often do look at mitochondrial barcoding markers. These guys have swapped their mitochondria extensively, and they're not necessarily doing it now. They did it in the past, and we can tell based on the barcoding genes that they are slightly different. And so it actually raises a huge number of questions on how did they evolve? How has hybridization impacted their diversification? And that's things we're working on right now. I don't have a good answer. All I know is that they diversified in this massive explosion that seems to also have involved mixing between species. And whether that promoted or reduced speciation is something I'm really interested in. My last sort of science question here is for the Borealis crassa inundata. So this is genomic data. And what you're seeing here is my genomic data for across the whole Borealis crassa inundata group. Now, inundata is supposed to be in the top end and crassa in the Kimberley, which are these yellows. And Borealis is here in just in the Kimberley. When I do this sequencing, what it shows me is actually across what is supposed to be crassa and inundata, it's all one species. There's gene flow between the region across the Victoria River District. For Borealis, all one species within the Kimberley. But these bars here, so every bar is an individual. And if a bar is fully blue, it's Borealis. If a bar is fully yellow, that individual is a crassa / inundata. But what we find is a bunch of individuals across the Northern Kimberley that are basically 50-50. So that means that each of these 6 individuals is actually a hybrid between Borealis and crassa inundata. And more interestingly, so I have mitochondrial DNA for these individuals, which is shown on this bottom here. And you get your mitochondrial DNA from your mother and not your father. And so what we can tell is these hybrids are 50% from Borealis and 50% from crassa inundata, but they all have their mitochondrial DNA from a Borealis mother. So this tells us that we have one-way hybridization between these species. So Borealis females can't tell the difference between Borealis males and crassa males, but crassa females appear to be able to. And I think this explains the difference in calls, because this huge genetic clade that is crassa inundata is different in the Kimberley. They call differently. And we think that this is a signal of reproductive character displacement, which is basically in the Kimberley, crassa inundata had to be different so that its females could tell who was who. So I think there's a lot of really interesting questions about how Uperoleia species arose and how they diversified, including their calls. So I'm just going to shift now just to a little bit of trying to catch frogs in the tropics. I'll say it's a pretty rough gig sometimes. You need rain to catch frogs. That shouldn't be very surprising. If you don't have rain, there aren't any. The problem is when there's too much rain, which means that either you can't get to where it's raining or where you want to go because it's flooding, or you have to run away from the place you want to be, because if you don't leave, you'll never get out. So this happened to me actually where this video was recorded in this particular storm in my last field trip, that we were trying to work in Lindman National Park and it was rain predicted. We're like, yay, we drove all day to get out near Newker in eastern Northern Territory. And then the rain was so much that we immediately turned around and left. So I'll just play this video. This is just what it's like frogging out night up in the top end. Hear your honks of the convex and your lovely laughing tree frogs and your lightning and thunder. There's a bunch of other things under there, but you can never hear them under those two species. So the other things that you have to do is wait and see where the rain, if it's going to be okay. So my last field trip, and everyone, I warn people that I'm going to do this, but they never believe me because it is genuinely terrible to do. So normally we start work at 7 or 8 o'clock at night and work for a while until we're done with what we want to achieve. But I say, okay, if it's raining really hard, we'll just go to bed, but then I'll wake you up when the rain ends and take you out. So one night we were out frogging and then the rain came and I wanted to go to a place that I wanted it to rain first. I didn't want to get flooded in. I wanted to see how much the rivers came up. And so I drove everyone home and put them to bed at about 9:30 at night. I said, okay, I'll just keep checking the rain and then I'll wake everybody up when it's done. And none of them believed that that was true. And then at 1:30 in the morning, I woke everyone up, said, get dressed, we're going frogging. To which I think they hated me. And I remember that night driving home at 4 because I'd had sleep that night. I never get to sleep at night. And everyone was very upset with me because I was, 4:30. I was like, I'm not even tired. I could keep going. And I think they could have just abandoned me on the side of the road at that point. The other hard thing is rain is never where you are. I probably drive back and forth across the Northern Territory 3 times each year chasing rain. And it's very difficult. And we're getting to the point that all the least studied places are now just the hardest to get to and also probably full of saltwater crocodiles. So it gets quite difficult to do that at this stage. So here's some photos. So this is the Kimberley I was very lucky to do some aerial work. And essentially you're trying to get from point A to B across these landscapes that are just, you can see that normally there's a river, but now that river is basically the whole landscape wide. It becomes very difficult to get to what you want to get to. You don't want this. This is full of saltwater crocodiles. You want these lovely isolated swamps. And when you're up there, the rain doesn't come down in like a big set of clouds. What you see is these isolated storms just sweeping across the landscape, and they dump immense amounts of rain in one go. And that makes your job harder because you literally need to be exactly where that happened. So you'll be driving and you'll say, okay, the rain literally just stopped exactly at this point, so I might as well turn around. So these storms follow a path, they're very specific. And for that day, if you're not in the line of where those storms are hitting, you actually can't find frogs. It is beautiful though. I basically never go up north in the dry season. And as far as I'm aware, it is a combination of fluorescent green, red, and blue. So it's usually not overcast. It's usually those spotted storms running across the landscape. And so I send these pictures to people who work on skinks and geckos. I'm like, man, it's so green up here, isn't it? Because they're like, what are you talking about? Where are you? So this is actually, if you've ever driven through Gregory National Park, this is what Gregory looks like in the wet season, bright green. That's just taken from the road through Vic River. This is a really typical example of some of the issues we have. So this is us trying to get to a private gorge on a property. And the landowner was super lovely. He lent us an ATV so we could, I think that's his head right there. So he took us out in the daytime to show us how to get to the gorge and all of those sorts of things. Super wonderful. So come back at night. And then when we went back at night, you can see us rolling in, and this tree is the boundary. This is an enormous storm about to hit the gorge. And of course, we're very happy about that because the frogs. But by the time we got to the gorge, it was fully in flash flood. We were physically watching the water go up. You could see it rising. And then we just had to turn around and go back because those gorges are quite dangerous in the rain. And so it's just a really typical thing, like just too much rain makes it almost impossible to do the work. But we do see lots of cool things. I do typically have a no touching of snakes rule, but if you want to catch snakes, you go look for frogs. Basically, frogs, I acknowledge, are snake food, and it's the best way to catch them. This particular snake was an enormous olive python sitting on the Stewart Highway, just north of Catherine, north of Catherine. And after we took it off, 3 different road trains went past. It's such a big snake, he could barely get it off his arm, and it had a grip, and his arm was starting to turn red, just to get it off the road to stop it from getting run over. But typically there's a no snake handling rule, but he works on snakes, so I trusted him with a python. My favorite thing about working up north is the birds at night. They are crazy what all the birds do. I mean, bustards just run at the front of your car, so do nightjars, but for some reason there's a lot of owls on the ground. So you just drive around, and I think it's because they're also eating frogs. There's always just owls. This owl was not flying around. We were just driving. We're like, what's that? Oh, another owl. You actually have to sort of turn off all your lights because they get too blinded to fly away, but it's that's just where they are. So it's a really funny thing. They're like snakes and owls all over the ground. It is, of course, very, very nice up there in the wet season. There's literally nobody there. There's so much water. All the waterfalls are amazing. It's my field assistant. I have another picture of him sitting under the waterfall, and he doesn't look happy. It's a really funny picture, but this was my waterfall down here, and that was his waterfall. And so it's actually just really incredible what you get to do during the day up there. And finally, of course, there are just amazing frogs everywhere. So this was actually just the frogs we found at Wildman River, which we made this for the rangers so that they could help ID the frogs that we found. But they're absolutely incredible. So, it's wonderful to go up there and find all of these amazing species. So I think that's my talk for today. Hopefully it didn't go too terribly long. If anyone has questions, I'm more than happy to take them. You can throw them through on chat as well.
Marvelous. Thank you so much, Renee. That's really great. We obviously can't do Frogs Big events without our speakers, and we thank you very, very much for offering your time and expertise for us all to enjoy what you know about Uperoleia, which is extensive and exciting. So thank you very much. We have an Andrew Isles book voucher that we'll send to you as well as way thank you, and know that we're really grateful and really appreciative and really enjoyed your talk as well. What I might get you to do, Renee, is turn off the recording, and we'll get some questions answered, if that's all right.